Hello everyone, this is Dr. Pooja and welcome to my channel. So, have you ever wondered where researchers obtain real next generation sequencing data for their bioinformatic projects? Whether you are performing RNA sequencing, metagenomics, whole genome sequencing, or antimicrobial resistance analysis, you need access to raw sequencing data. Fortunately, the NCBI provides one of the world's largest repositories of sequencing datasets completely free of charge. So this source is called the Sequence Read Archive, that is SRA. So in today's tutorial I will show you exactly how to search, explore, understand and download sequencing datasets from the NCBI SRA dataset database. Okay, let's begin with the basic. So before we proceed, what is SRA? So it stands for Sequence Read Archive and it is world largest public repository of raw next generation sequencing data. Unlike databases such as nucleotide or protein, which store processed sequences, the SRA database store the original sequencing reads generated directly by sequencing instruments, and these raw reads have not been assembled into a genome or fully analyzed. The researchers use them as the starting point for bioinformatics workflows. Now, the SRA contains data sets from a wide range of studies, including whole genome sequencing, RNA sequencing, metagenomics, single cell sequencing, transcriptomics, epigenomics, and microbiome studies. Because the data are raw, researchers can analyze them using different pipelines. Now, actually, the SRA it is widely used because it allows scientists to reuse publicly available sequencing data without having to generate expensive new data sets. Now let's move towards how to search. Let's go to the official NCBI website. And select SRA from the database drop-down menu. And click on search. Now we are ready to search for sequencing datasets. So for this demonstration, we will search for an RNA-Seq dataset related to biofilm formation in Pseudomonas aeruginosa. So I am typing here Pseudomonas aeruginosa biofilm RNA-Seq. And just click on search. The research results display experiments that match your query and each representing a sequencing project contributed by researchers around the world. On the left hand side, you can see the access for that particular research or project, then source, either it is from DNA or RNA, then library layout. So that library layout it is also important because it highlights the single end and pair end. So the library layout indicates whether sequencing was performed using single cell sequencing, where one of each DNA fragment is read, or pair end sequencing, where both ends are sequenced. So pair end reads generally provide better alignment and accuracy and genome assembly. Then there is a platform name, it is given here. So the sequencing platform tells you which machine generated the data it is. So common platform include WGS, then Illumina, Iron Torrent, Oxford Nanopore is there, PacBio is there. So each platform produces sequencing rates without different lengths with different lengths and error characteristics. Then if you go on further, so it is given strategy, that is the isolated is of a genome or the data of RNA-Seq or other, right? Then FASTQ files are there, BAMs are there, which file type the data it is. Now if you see here, I'm going to click on one project. So you will notice several accession numbers associated with each dataset. You can see here SRX 3385796. So this is one accession number which is represented as SRX. So what exactly it is? So there are four types of numbers, that SRP, SRX, SRR and SRS. So SRP refers to the sequencing project, then SRX it refers it represents an individual experiment, while SRS identifies the biological sample, and SRR it is correspond to the sequencing run which contains the raw sequencing read. And when downloading sequencing data, the SRR accession is usually one you will need. Okay, if you can see here on the last SRR and the number it is given. So if you can see here, you will see the detailed information, the external ID it is provided here, which organism it is, what kind of sequencing it is done, that is RNA-Seq, which instrument it is used, that is Illumina, NovaSeq X Plus, then runs, and the bases, and how much download the number of data it is provided. Then the external ID it is also given, then it whom it is submitted by, that is molecular biology faculty of pharmaceutical sciences likewise, then which study information it is provided, GEO accession number it is given, and then again you can see here library it is given. So in the library you can see the layout, that is paired or selection it is complementary DNA, then which kind of source it is, transcriptomic, then strategy it is RNA-Seq, and again the instrument it is used. Then construction protocol it is also given here. Now the metadata describe the experiment in details, and this information often includes the sample type, tissue or organism, growth conditions, collection location, sequencing platforms, etc. So always review the metadata before downloading the data, because it explains exactly how the experiment was conducted. Now how to download the FASTQ file. So if I am going to click the accession number, that is SRR, it will take you to the another page, and you can see here metadata, then analysis, then reads, then data access, and the last one it is FASTQ download. So you just click on FASTA or FASTQ download, and then from here click on FASTQ. It just downloaded the FASTQ file. So depending on the data set, downloads may include the single FASTQ file for single end sequencing and two FASTQ files for pair end sequencing. So this is all for the today. So let's quickly summarize what we have learned today. So we learn how to search sequencing dataset, interpret SRA accession number, explore experiment metadata, identify sequencing platform, and download FASTQ file. Okay. So if you found this tutorial helpful, please like the video, subscribe to the channel, and share it with your classmates and research colleagues. Take care and bye bye, and thank you for watching.